So you've all seen the paper. We're very excited. We were very excited to see this. Of course, it very relevant to the work we do, as one of the molecules that we're working on was actually in the paper. So without any further ado, please go ahead, Dr. Gersch.
Well, thanks, Spring, for the introduction and for the opportunity to share the work. So really, I'm presenting here, but the work was really done by 4 fantastic people who've all joined here, and so I'm looking forward to be able to share this, but also to discuss this then with you and with the team later. So what I thought I'd do, I put together a few slides on the work that we did, but really at the beginning zoom out to get everybody on the same page, because we're interested in the ubiquitin system, where these isopeptide bonds are made and where ubiquitin is translated into all sorts of outcomes. The outcome that's particularly relevant here is autophagy, or in this case mitophagy, that we're looking for. And these processes get antagonized by dubs, because they can cleave these bonds here, both in the chains as well as the bond between ubiquitin and the substrate. The basic idea of this concept of dub inhibition is that if you inhibit the dubs, you basically supercharge ubiquitin-dependent signaling. You take out the antagonizer here, and then there's more flux into this direction. If it's a dub that has a specific substrate, then you would do this for a single substrate. But if you have a dub like USP30 with a lot of substrates, then there's also a lot more flux in this direction. And so, of course, the challenges to do this are that you need potent and specific molecules, and you also need to know how the dub recruits the substrate, because all the modulation that you do for the biological pathway is going through this substrate. So in terms of the molecules, when I gave my application talks to become independent, the time was a very different one, because there were some molecules out there, but the higher quality ones were just emerging, and I'm really amazed at the progress that the field has seen in the last 6 years or so, with more and more specific molecules coming up and entering the clinic. And so there's 2 ones that I want to highlight. The first one is on USP1, where this compound, for example, has completed phase 1 trials. Been hit some issues. The Tango project has been put on hold, and for KSQ there's also been some issues detected. But the efficacy in the preclinical models, at least, so this is a mouse model where inhibition of USP1 as well as PARP inhibition leads to complete shrinkage of the tumor. This is quite staggering, and I think it just shows the potential of dubs as targets. And the second program is the one, of course, that I don't have to here preach to the choir, is USP30 for both Parkinson's as well as chronic kidney disease. Just want to highlight this data here, which came out of this very recent study, because USP30 was connected, as you will all know, to Parkinson's due to the genetic link to juvenile Parkinson's patients, and not necessarily to what clinically is considered a hallmark of Parkinson's, as in a loss of dopaminergic neurons, for example, here, which can be artificially triggered. So this loss of dopaminergic neurons does not occur when, in this case, these mice have been fed a USP30, a brain-penetrant USP30 inhibitor that Mission Therapeutics makes. And I think for me, this was data highlighting to show that there is not just a genetic link there, but there may also be a functional link, even though it's, I think, still unclear how particularly dopaminergic neurons are rescued by USP30 inhibition. Right, so let's talk about this process which led to the identification of USP30 as a drug target. And so this is this process where defects in mitochondria are sensed through stabilization of the kinase ping1, which then leads to recruitment parkin through phosphoubiquitin, and the second step phosphorylation of parkin, which then in a feed-forward mechanism leads to more and more parkin coming to mitochondria, specifically those mitochondria that are damaged, and then ultimately separation from the mitochondrial network and mitophagy. And the USP30 is the only DUB that has a transmembrane segment anchoring it on mitochondria, as well as peroxisomes. And so you can think of USP30 being able to antagonize this process in two different ways. Firstly, upstream, so by directly deubiquitinating certain mitochondrial outer membrane proteins. And so this would decrease the substrate that PINK1 has available to put phosphoubiquitin onto mitochondria. And then secondly, also once this mitophagy pathway here is kicked off by antagonizing these chains, and you will see data later on this second part here. The data that basically implicated USP30 as a negative regulator of parkin comes from Genentech, where they showed that USP30, if depleted, or if USP30 is depleted, there is a lot more mitophagy ongoing here, measured with this mitochema assay. And so this is under basal conditions. Each of these spots here is a site of mitophagy. And so this implicated USP30 as a negative regulator. USP30 does a lot of things. It's, I just mentioned basal mitophagy, so without global depolarization. We know that it interacts with components of the TOM complex and primarily localizes there, and so also regulates mitochondrial import, morphology, abundance, and there's also big connections to cell death. And so this is really the work of a lot of labs that have looked into what USP30 does functionally. But really for this for our interest, this ability of depletion of USP30 to prevent the loss of dopaminergic, again here measured in a different way, where these the dopamine here is quantified via the stain, and so this is preserved in USP30 knockout mice, which also without the alpha-synuclein expression already have higher levels of dopamine. As you can compare here, this is really motivating for small molecule means of mimicking this condition. And so put together is everything is that Parkinson's disease is characterized by loss of dopaminergic neurons. We know a connection to mitochondria through the genetic risk factors through this pathway here, of which USP30 is a negative regulator. And so if we're able to inhibit USP30, we may be able to generate therapy for Parkinson's disease. In terms of the compounds that we looked at in this project, we looked at two compounds. One of them, this compound called 39, which is very, very potent both in vitro as well as in cells. The specificity data you will see in a second. And we were happy to work with this compound because there was a study that I'll also show in a second that benchmarked this in various cellular assays. So we knew that this is acting on target in cells. And then just this year, this compound here became available or became public, which is a covalent USP30 inhibitor by emission. It's their brain penetrant version with this alkylated amide here. Also very, very potent, very, very potent in cells as well, and also with really, really high specificity. Voila, where are we now? Right. So it's really this remarkable specificity here. If you have a compound that inhibits in 2 to 20 nanomolar, and you can inhibit, or you can incubate other DUBs with more than 100 micromolar, and you don't see an inhibition, this is really, really striking, because we have about 100 DUBs in the human genome. All of these DUBs process ubiquitin, meaning they have to some degree or another the same substrate. And then still you have a compound here that is able to select one of them over the others. And so this I found very, very interesting and very intriguing. This is the data that I just mentioned, which is a DUB panel where this compound 39 at the concentration of up to 100 micromolar was assayed, and USP30 really, even at this very high concentration, was the only DUB measured. And so this really resonates with the specificity in cells that was assessed by Benedict Kessler's lab. Benedict's lab also did some further analysis here. They mapped the binding site through hydrogen deuterium exchange mass spectrometry, which is really, really accurate compared to the binding site that we saw. So the responsible residues were mostly identified. So this was really a great example for how HDXMS can be used to map compound binding sites. And they also concluded that there may be what they called a pseudo-covalent binding mode, because this compound here has a really, really slow off rate. So it interacts with the protein very, very tightly, and this explains this incredible affinity. And so to summarize, there's a clear, there are potent and specific inhibitors. And so that's really, really exciting for DUB. And also the clinical rationale is clear, and was clear enough to have motivated clinical trials for both acute kidney disease and Parkinson's. And so the question that really wanted to address is how do these compounds actually achieve this specificity? This is also, if you compare to other available DUB inhibitors, really, really remarkable. And so this meant that we had to get structural information of the compound bound to the protein. For the purpose of this talk, we'll focus here on the non-covalent compounds, but we'll have a little bit of a comparison later with these covalent ones. And so I knew this wasn't going to be super easy, because during my postdoc, I was working with David Commander and was able to solve this structure of an engineered human USP30 in complex with K6 diubiquitin. That was also a project that took about 3 years and a lot of constructs and a lot of optimizations to complete. And the details are described here in the manuscript. But what we had to do to get to this construct here was that we had to remove 2 areas that were measured to be flexible, actually also by HDXMS. And then also these deletions were not enough. These deletions gave some protein, but the protein was incredibly insoluble. And so you may remember that this is the times before AlphaFold. And I remember we had to do a lot of alignment searches and manual homology modeling. And it was basically a wild guess that we said maybe one of these deletions here exposes an area on the back that is hydrophobic. And if we mutate this to be hydrophilic, we may get somewhere. And this was the actual moment that then made us be able to progress, because only these mutations made the protein soluble. We worked very hard to get the human protein there, because we knew that for translational reasons, the structure of the human, or even the construct of the human protein, was very, very useful. We had competition from a lab that did a similar structure here in zebrafish, and so the binding mode was the same, but they didn't need any of the engineering. And so when we started in my lab working on this, we tried this construct here. But didn't get anywhere. And so that also makes sense, because there's still a lot of loops here that are flexible, and parts of the protein that are not well resolved. And so the basic idea that we had was that how about we retain some parts of USP30, but we add elements of DUBs that did crystallize. And so we took various DUBs. I'm going to show you here the version that worked, that has USP14 in the fingers region. So there's crystal structures of USP14 with compounds. So we figured we could use this area here. And then in the back, there was a little loop here that was actually added to this engineering, which in the HDXMS still had a very, very high uptake of deuterons. And so there we engineered a part of USP35 in, because that's the only DUB that had a secondary structure there, and we figured that this would stabilize. And so all of this was enabled by parallel AlphaFold modeling to verify our designs. And Nafizul, who did the work, went through a number of these iterations. I remember the lab meetings usually had a decision tree of this construct and this construct and this variation, and this didn't crash, this didn't crystallize, or this crystallized but couldn't be optimized. So there was a lot of work along the way. But ultimately, what worked was this construct here that I show you down here that has two areas from USB 14 and 35 and is USB 30 in any other area. And so we characterize these chimeric dubs in a variety of ways. We made sure that they still bind ubiquitin. So this is a ubiquitin probe assay where the protein still shifts. You can see it's a slightly smaller than this construct one here. Still binds ubiquitin. It's still stabilized by the inhibitor, and it's also more stable. The inhibitor complex is more stable than the C1, and that for crystallography usually is a good indication that something is more crystallizable. And we also measured the ability to inhibit USB30, and we realized that if you compare these two constructs, the inhibition is roughly the same, meaning that we are engineering the protein, but we're not touching the sites that are responsible for recognition of the inhibitor. And so for the inhibitor, we started to work with this compound 39, but for crystallography, we need rather high concentration. The protein, the compound wasn't that soluble, and so at some point, Nico, who was the chemistry lead here, came up with the idea to use a version that we had made for a completely different reason, but behaved much more soluble, that had a hydroxyl group here. And so this decreased potency by about an order of magnitude, but it made us, or it enabled Nafisol to reach the concentrations that were needed for the crystallography. And so just to clarify this here, we're not pitching this as an improved compound to be used for any purposes, but it was just for technical reasons the compound that we used for the crystallography. And so this is then culminated in this structure here. I should say we went through quite a number more chimeric concentrations, but these were the constructs that were actually made and tested. Once we had the crystal form, also that required multiple rounds. Additive screening was particularly valuable there to push the resolution, and then for the data optimization, we also had to do various things. So this is basically a lot of rescue and backup strategies in one project. But the person who really pushed this through was Nafisol. Chemistry was done by Nico, and the cellular validation that you will see was contributed by Kai and Jan. So a real team effort here. Right. So the compound you see on the left. Maybe let's just compare first to this ubiquitin bound state. So this is the catalytic domain here, where there's a catalytic site down here. This is this cleft that recognized the ubiquitin C terminus, and this is what's called the S1 site, where the globular part of ubiquitin is recognized. And if you do a superposition, you can see that the compound binds right across this cleft here that normally guides the C-terminal tail of ubiquitin to the active site. And so inhibition is facilitated by various mechanisms, but one of them is blocking ubiquitin binding. Now, if we look a little bit closer here, we can see that we have good density to resolve all three parts of the compound. I show you this for one chain, but the binding mode is the same for both chains that we have in the asymmetric unit. And we're fairly certain about the positioning, even though the resolution is where it is. We're fairly certain about the positioning of all the atoms, with the exception of this hydroxyl here, which is not recognized and which we can't position well. You can also see that it's the molecule is almost engulfed by the protein, and so there's a lot of surface that contributes to the binding, and so this really contributes to the covalent binding mode that the Kessler lab proposed here. We zoom in a little bit, we can see the different structural elements that will become important for the discussion of the binding mode. So here, this loop here is called the blocking loop, that houses one of the catalytic residues that binds around the compound here and wraps around this fluorophenyl moiety. There's the blocking loop 1, which normally recognize a hydrophobic residue of this globular part of ubiquitin. And then above this cleft here, there's a loop that's called the switching loop, and that's called switching loop because in USP7 it switches between different conformations in the apo and the ubiquitin bound form, and you will see that there's also quite some switching happening here in USP30. Now the different parts of the inhibitor are all recognized. So there's a couple of polar interactions here that we highlighted in this figure. You can see of these two central amides here. There's really only one hydrogen bond acceptor here that is not specifically engaged. All the others are engaging with the counterparts in the protein backbone. That probably also explains why any modification in this area from what we could see in the patterns is not tolerated. The sulfonamide here is also engaged not only by these amides here, but also because it's at the tip of a helix, so the dipole will open of the helix will pull this in, and so these are the polar parts. And then you will see that if we can go through these three hydrophobic parts of the molecule. If we first look at this fluorobenzyl moiety here, you can see that there's a hydrophobic pocket that I'll talk about separately. We can now move to this what is basically a phenylalanine in the molecule. You can see that there's a hydrophobic, not really pocket, but at least cleft here. It's also larger in the Kluge paper from Mitophagy. There's some cyclohexyl structures as well, and so this is perfectly conceivable that some larger structures would fit here as well. And so what was then quite exciting for us was that this sulfonamide portion of the molecule then really ended up somewhere where we didn't see this or we didn't expect this to end, because this is a pocket, as you will see, that doesn't exist, but it wedges in to a part of this domain here with a lot of other residues. And so I'm not going to discuss the residues now. I'm going to discuss them later when I show you the mutation. But this is just to say that all of these areas are closely recognized. And you can see from this base representation that large parts of the molecule are surrounded by protein residues. So to talk about this fluorophenyl moiety down here, if we overlay this now with the ubiquitin bound structure, you will see that this is the part of USP30 that normally recognizes leucine 73. And so leucine 73 in ubiquitin is a very, very important residue because this basically anchors the C-terminal tail in this cleft and then allows for the rest of the tail to find its way to the active site. And so we know that this is important because if you mutate leucine 73 to a proline, you make ubiquitin non-deconjugatable. So basically you prevent all DUBs from acting on this. So this is a very, very critical residue. And so it also makes then, from this point of view, perfect sense that a DUB inhibitor would block this site here by fitting right in. This is this part here. And then if we now look into this part where the fluorophenyl, where the sulfonamide goes, we have here this same overlay of USP30 in this diubiquitin bound conformation. So this is the structure in green, and we also have a merge to the compound bound structure. And if I play this now, you will see how if the compound adds in, there's a few small conformational changes down here, but the largest conformational changes really occur here at the switching loop. So you can see this helix here forming from residues that before were extending to this other side. And you can also see conformational changes because I'll play this once again. If you focus on this phenylalanine over here, this moves over and it basically displaces a tryptophan that moves out of the way. And so this this orientation of the phenylalanine taking the place of the tryptophan anchors this newly formed helix on the other alpha-5 helix here. You can also see in the back, I'm going to play this one last time. If you look at the back, there's another phenylalanine there. And so this rotates out, and by rotating out, it opens the space for the compound to move in. Also because this alanine here that's hidden behind this is also moving over and sort of opens the pocket. So to summarize this, if you compare this to ubiquitin-bound form, this phenylalanine here moves to the back. This alanine moves to the front together with the helix, and this opens a pocket, which is then accommodated by the compound. This compound is then contacted also by this new conformation of the switching loop, which is held in place because the phenylalanine that normally is part of the proximal ubiquitin binding site is anchoring the switching loop on the helix. So quite a lot of movements. We do not have an apo structure of USP30, so we can't say whether these are truly compound-induced changes, because we can't do a clean comparison to the apo form. But all of these modes are incompatible with ubiquitin binding, we believe that these are true compound-induced conformations. And so I started this part here by saying that we're interested in the specificity. And so we, of course, then looked what actually explains why these compounds are specific for USP30. And two residues stood out to us. One of them was this leucine here, which in all other USPs is a phenylalanine. And the second one is this phenylalanine here, which in all other USPs is a tyrosine. And these two residues are at the center of the binding site. So this is an overlay of the structures of these dubs here. You can see that this additional hydroxyl group of what normally here is a tyrosine would clash with the central diamide. And we also have this leucine here, which is a phenylalanine in the other USPs, which would not allow this fluorophenyl to occupy this site. We then did the obvious experiment of mutating these residues. For the F2Y mutation, you can see here that this generates more than 1,000-fold difference in affinity. So it decreases potency on USP30. We couldn't do the same experiment with the L2F because the L2F also didn't recognize ubiquitin very well. But what we could do was we could measure protein stabilization by compound 39. You can see that the wild type is very strongly stabilized, but these two are less so, meaning that they bind to the compounds less tightly. This is done at above the IC50, so that explains why there is some stabilization measurable, but it explains or it confirms this hypothesis that these two residues that are specific to USP30 also confer specificity for the compound. We were then asked to confirm that these compounds actually work in cells and also that the binding mode is done in cells. And so what we adopted here is this relatively dated but still beautifully working model where HeLa cells are constitutively overexpressing YFP-TAC-Parkin, and we can then look at protein ubiquitination by using a tool that the lab of Mark Hofstrasser published recently, which is a very high affinity ubiquitin binding domain. We can basically do a pull down with this and look at what proteins are ubiquitinated if we plot for the particular protein. So this is data here showing that TOM40 here is typically not ubiquitinated. If we have global depolarization by CCZP for an hour, we can see mono ubiquitination. But if we do this with cells that were pre-treated with USP30 inhibitors under conditions where you expect USP30 inhibition, we can see a lot more ubiquitination happening, which could be multi-mono or poly ubiquitination. And the same is also true for TOM20, which is this more often studied substrate of USP30, where you also see mono-ubiquitination and a little bit of poly or di-mono coming up under depolarization. And with USP30, this process is just much further ahead, similar to what you would see if you waited longer. It's noteworthy that this is not true for all mitochondrial proteins. Mitofuscin, for example, does not respond to USP30. And so whatever USP30 is doing to regulate mitophagy, it's not by globally antagonizing Parkin, at least under those conditions, really likely related to the TOM complex, where accidentally also pink one is located. So in a way, this makes sense. And so the cellular validation we completed here that these compounds actually do inhibit cellular USP30, but we also wanted to confirm that this mode of inhibition is true in cells. And so for this, we overexpressed wild type or this F2A mutant, and then looked whether the protein is able to recognize this ubiquitin binding with this ubiquitin probe here. The wild type did, but not if cells were treated with inhibitor, whereas with this compound resistant mutant, you could pre-treat the cells with the inhibitor and then still see this adduct forming, indicating that this residue is also important for mediating this inhibition. Some data that's not yet in the manuscript is that it's not only about these residues down here in this fluorophenol that are important. They're important and specific, but there's also various other residues in this upper part here that are important. So here we looked at this isoleucine 154, which basically caps this top hydrophobic part here. We also looked at this phenylalanine here, which is the one that did the big move. And we also looked at this alanine down here, which is the one that moved with the helix. And we can mutate these three here, and they also confer compound resistance. Not as strong as an effect as this F2A, which was pointing right at the compound binding site, but just to say that there's additional residues around them. These two are also USP30 specific that are helping to confer specificity also in this other part of the molecule. And by looking at this phenylalanine, this is particularly striking because it's actually not contacting the molecule directly, but it's the one that anchors the switching loop in this conformation that is compatible with the compound binding. And so if we now focus on this phenylalanine here, this is a good way to compare the structure that we solved with a structure of a covalent USP30 inhibitor that scientists from Camor and Amgen deposited in the PDB a while back. I should say that this construct or this structure here was generated by, or this structure includes an additional antibody fragment that stabilizes USP30 down here. The construct is the one that I was talking about earlier. That's the one that was used for the diubiquitin structure. And so if we now overlay this, we can see that both of them bind in a similar area of the catalytic domain, but really touch different areas of the domain. If we zoom into this the cleft here, you can see a lot of residues. Some of them you're familiar with. This is more parallel. This is still in the middle of the binding site, and you can see this covalent linkage over here. But what's really striking is that there's this additional phenylalanine in here, which is actually the same one that I was just talking about. So if you now make an overlay where you look at here, this is this extended phenylalanine in the ubiquitin-bound form. This is the phenylalanine in the state that we show. And this is the phenylalanine in this covalent state. If you overlay them all, you can see that there's three different conformational states of the switching loop that are really distinct, one for ubiquitin binding, one for the covalent compound, and one for the non-covalent compound. And that really highlights the conformational plasticity that this region here has. And it also highlights how distinct these binding modes here are. Right, so this was comparison to other USP30 inhibitors. I'm not going to go into much detail comparing this to a lot of other inhibitors of other USPs, but I just want to show you that if you overlay them, there's actually one side where a lot of the compounds overlap, and this is this ligandability, what we termed ligandability hotspot, so the pocket where leucine 73 of ubiquitin is recognized. And this was interesting for us because once we started to look into these comparisons, we realized that there's actually two other compounds that were made for USP7 as well as USP14 by different people with different rationales, different ways of converging in a binding mode where this para-halogenated phenyl group here basically adopts the same position as the parafluorophenyl of USP30. And so these overlays here of the USP14 and USP30 structure, or here of USP7 and USP30 structures, these were made based on the protein residues alone. So we didn't force the chemical compounds to be as similar as possible. We just overlaid the proteins and realized that this meant that the compounds would also emerge. And so this really made us suggest that maybe this is a bit of a privileged scaffold for DUB inhibition that is able to bind this site that is so critical for DUB activity. And if we spin this out, we could conceptualize this, think of this here as an anchor that binds where leucine 73 binds, basically this being an anchor, and then have the molecule extend into different areas. For USP30, it would be this cryptic pocket down here, as well as would be this pocket towards the blocking loops. We can now annotate also the structures of these inhibitors with these different parts here. So where you would have a pocket being occupied by this part of the USP30 inhibitor, you would have another pocket that's occupied by molecules that bind towards the blocking loops. Here, here. Again, widely different chemical matter, but the sites are similar. Would have elements going towards the catalytic cysteine. You would have this pocket here and the USP28 inhibitors under the helix, so on the other side, or you could have, again, widely different chemical matter going to the same sites, in this case here towards the S1 site in USP7. And all of them share some chemical element, either similar or not so similar, that occupies the same hotspot area here in the 73 pocket. So with this, I think what the study shows is that these non-covalent USP30 inhibitors occupy an induced and cryptic pocket that's not visible in the substrate bound state. That there's residues that are specific for USP30 that allow these compound-induced conformational changes, and these conformational changes of the switching loop then underlie the specificity. We see this from the mutations, but also if you zoom out, this may be the beginning of a logic for DUB inhibition that may allow much more rational approaches to finding and improving compounds if we have a framework to understanding them. And so this we did this all with a focus on USP30, but I just want to end by saying that this is of course been facilitated by all these protein engineering efforts, which as a strategy that is not necessarily restricted to USP30, but could be used for other protein classes or other DUBs and other inhibitors as well, meaning that there's really been some addition here on the method side as well as on these insights on USP30. So with this, I'd like to thank again the team. Nafizul is the true hero who made these constructs, profiled them, crystallized them again, and crystallized them yet again, who made this project really possible. Nicholas did the chemistry, Kai and Jan did the cellular validation, and of course we've been helped by many other colleagues in the lab, as well as various collaborations. Lastly, I'd like to thank the various types of funding that the labs received that has allowed us to pursue this really challenging project and to bring this to where we are now. And with this, I'd like to thank you again for the invitation to present here, and I'm very much looking forward to the discussion now.